So I'm very pleased to introduce Dr. Gustavo Turecki. I just knew that he's a student from this university. I'm very proud to know that we have such outstanding students coming out from this university. He's a professor of psychiatry in human genetics in McGill University, Montreal in Canada. And he's going to talk about the early life environment, adversity, genomic plasticity, and psychopharmacology. So, thank you.
Bom dia, a gente tem aqui avança e descende. Você tem 30 minutos, eu aviso quando faltar 35 minutos, eu aviso quando faltar 5.
Ah, tem 35 minutos?
Porque são 5 perguntas.
Ah, tá já. Okay. So, hello everyone. Oops. So it's really a pleasure to be here. It's always nice to be back. So what I like to talk to you is it's about the work that we've been doing, trying to understand how is that the early environment can modify our experience, and that, or how is that negative experience that we have in early life can modify our behavior, and what sort of molecular processes can explain the behavioral modifications. So, oops, so if I, okay, all right, sounds good. Yeah, okay. Okay, very good. So the work that we do, it's focused on suicide, and we try to better understand what are the different mechanisms that make people die by suicide. So one of the things that we know is that suicide, it's complex, as most of the psychiatric phenotypes, and when we look at the different factors, we group them in factors that act more distally, and then factors that act more proximally. So, the distal factors would increase predisposition to suicide, whereas the proximal factors act as precipitants of a suicidal behavior. Now, among the distal factors, over the last 10, 20 years, we have learned a great deal about how biological factors play a role, how genetics play a role. And one of the things that we have been interested over the last few years is the role of early life adversity. So we know that most of the individuals who die by suicide do not have a history of early life adversity, but a significant minority does. And one of the things that we are interested is in trying to understand how is that these negative experience in early life can increase risk of suicide much later on. So what I would do with you today is basically the first minute is just review a little bit of the data that suggests that negative early life adversity or negative early life events increase risk of suicide much later on, and then after, we'd like to review or discuss some of the molecular mechanisms that may help us understand these relationships. And then if I have time, over the last few minutes, I'd like to speculate a little bit how is that these molecular modifications may lead to behavioral changes that can then increase risk of suicidal behavior. All right, so there's lots and lots and lots of data that indicates that individuals who were exposed to adversity early on in life are more at risk of manifesting suicidal behavior later on. So this is data from a New Zealand cohort run by David Ferguson, looking at how events during childhood, and here you have sexual abuse and here physical abuse, then increased risk later on at the age of 16, 18 of manifesting suicidal behavior. And you see that there is a relationship between the type of adversity early on and the likelihood of manifesting suicidal behavior. So here as suicide attempts. This is from our own work in Quebec, where we have been investigating a cohort that's representative of the general population. I'm going to talk to you about this later on. And then here again what you see is that individuals who were exposed to physical and sexual abuse during childhood, and often, as is usually the case as well, a combination of both, when they grow up, and here's at the age of 21, they are more likely to present relative to those that were not exposed, they're more likely to present both ideation as well as attempts. And this is different data, is retrospective data from individuals who have died by suicide, and this is from the psychological autopsy studies that we have been conducting, where you see that again, individuals who die by suicide are more likely, above and beyond those that psychiatric controls, of having a history of early life adversity than as well compared to community controls. So what we know is that it's not the abuse per se, but rather the psychological experience of the abuse that may play a role in increasing this predisposition. So here you see that in the same cohort from Quebec, the identity of the abuser plays a role in the, moderating the relationship between having been abused and manifesting suicidal behavior in such a way that when individuals were abused by immediate relative members, then they are more at risk of manifesting suicidal behavior as compared to those that were abused by strangers. So really suggesting that something happens in the development of attachment or any other processes related to psychological development that plays a role explaining this relationship between abuse and suicidal behavior rather than the process of the abuse per se. So what really I think I find fascinating is to think how is that events that occur very early on in life and impact the life of these individuals, and what sort of mechanisms explain this process. So what I like to discuss with you is how is that we can think that, and at the molecular level, what is the changes that may help us understand this lifetime increase in suicide risk. So what we need to think is the brain. Yes, our brain is plastic, isn't it? And we respond to experience. So now what we know is that the brain is constantly responding to experience and adapting to that experience. Now one of the things that we know is that the capacity of the brain to adapt to experience is not equal through life. So there are periods in life that we refer to them as sensitive periods in which our brain is more receptive to adapt and to learn from that experience. Yes? So there are a lot of different examples in terms of brain function that are regulated by sensitive periods. So acquisition of language, for instance, is a very intuitive one, isn't it? All of us, we know that we are better capable of learning language early on in life, and having or not having accent, it's dependent on the age in which you learn a language. So for instance, our brain is initially totipotent to acquire language, but as it acquires one language, it decreases capacity of learning another language, as exemplified in this study here, in which by 6 months, really the brain shuts down to the total capacity of acquisition of perfect speech. Another intuitive example is acquisition of music. Yes, so we all know that if we learn music early on in life, we're better able to have perfect pitch and so on and so forth. and then the brain also responds differently to the experience of music. So there are studies, in imaging studies, showing that the brain not only is functionally but also structurally different when you look at individuals who learn music early on in life as compared to later on. So and there are a number of other examples in terms of brain functions that are regulated by sensitive periods. What we have learned more and more is that behavioral development is also ruled by a sensitive period. So this is data coming from animal studies that show that the early life experience is critical in shaping up behavioral development in rats. So for those that were yesterday at the course, I talked about the work by Michael Meany, in which looking at variation in maternal behavior, in naturally occurring variation in maternal behavior measured by licking and grooming, defines the development, the behavioral development of the offspring of the mums, and in such a way that the more the mum stimulates the offspring, the more exploratory behaviors the offspring have, and whereas the less the mum does, then the more inhibited the offsprings are. And this is translated at the molecular level by changes in levels of glucocorticoid receptor in the hippocampus, in such a way that the offspring of mums that are more stimulating have higher levels of glucocorticoid receptor in the hippocampus as compared to the offspring of the mums that are less stimulating. And this is stable over life of this offspring, and is really dependent on maternal behavior, because when you cross-foster the offspring in litters from moms that are less stimulating, then you completely reverse this phenotype, as well as the molecular marks. So what we've learned over or began to learn over the last 10 years or so, is that epigenetic processes are critical in the regulation of these functions according to critical periods. In other words, that epigenetic processes play an important role in the shaping up of biological functions in critical periods. So epigenetic processes, and we talked a lot about them yesterday, are of many different types. In particular, what I'd like to discuss with you today is one of these epigenetic processes is known as DNA methylation. So DNA methylation is nothing more than the addition of a methyl group to specific sequences in the genome, and particularly to CPG sites. And so the central dogma of methylation, or so as it has been until now, is that basically methylation in regulatory sequences like promoters play an important role in decreasing the activity of that gene. Yes? Now, we discussed yesterday as well that methylation in gene bodies are associated with increased function of the gene, but in particular what I'd like to discuss with you today is data focusing on regulatory sequences of genes, and where methylation acts primarily by decreasing, either shutting down the gene or decreasing the activity of the gene. So going back to the example of the rat, basically what we've learned from subsequent work that Michael did with Moshe Schiff, is that the maternal behavior in the rat, basically what determines is the methylation patterns in a promoter of the glucocorticoid receptor in the hippocampus. So the glucocorticoid receptor is a complex gene that has 11 untranslated exon 1 versions that basically determine the tissue specificity of the expression of that gene. So what they found is that in the promoter, so in the sequence that regulates the activity of this gene in the hippocampus, there is a major difference in patterns of methylation according to maternal behavior, in such a way that the more the mother stimulates, the less methylation there is in this particular sequence here, as you see here, and the less the mother, the mama rat, stimulates its offspring, the more methylation there is. And in such a way that you almost have complete methylation here. And this is important because this is an area where a transcription factor that drives the activity of this gene binds, and by having a lot of methylation here, you basically prevent this transcription factor from binding here, and therefore you prevent this gene from being activated. Yes? So basically, again, this is determined by maternal behavior. When you cross-foster, when you take the offspring from a high Lichen groomer and you put in a low Lichen groomer litter in the first week of life, you completely reverse these effects. Yes? So, so this was a proof that early life environment determines how genes are regulated by methylation patterns. So a lot of what we have been learning over the last years is that genes that act at the level of the HP axis are regulated early on in life through environmental factors and through early life experience. So I don't have to review with you what the HP axis is. You know, this is an axis that is important for psychiatry, so I imagine that you're very well familiar with it. But just to say one point that I think is important is that the HP axis, you know, besides the hypothalamus, the pituitary, and the adrenal gland, it also has a number of other areas in the brain that provide feedback to the HP axis, and among those areas, one which is particularly important is the hippocampus. So the hippocampus has a lot of glucocorticoid receptors that regulate the tone, the activity of the HP axis. So the HP axis has been investigated with relationship to suicidal behavior because evidence of overactivity of the HP axis suggests that individuals that do have overactivity are more at risk of dying by suicide. So this is, for instance, work by Bill Coriel doing a 15-year follow-up of individuals that were admitted to a psych unit and were tested with a dexamethasone suppression test. And those that had a non-suppression over a 15-year follow-up had almost had about 26% increased risk of dying by suicide. This is another evidence looking at individuals treated with corticoids in OPDs in first-line levels for many different causes, and where you see is that those compared to those that were not treated, and this is almost 1 million observations, so it's really a very large study that was published last year, looking at those that were treated are at increased risk of suicidal behavior as opposed to those that were not treated. And I could spend here hours discussing with you the evidence suggesting that HP axis dysregulation leads to an increased risk of suicidal behavior later on. So we were interested. Oh, I'm having a hard time here with this thing. All right. Okay, so we were interested in investigating whether or not individuals that were died by suicide and had a history of having been abused had different levels of activity of the glucocorticoid receptor in the hippocampus by methylation. Because, you know, I told you that early life adversity, it's an important risk factor for suicidal behavior. I told you that the early life adversity may, or early life environment, sorry, may regulate the activity of genes through methylation. So, and I told you as well that the HP axis increased risk of suicide. So that seemed like a good idea to look at whether or not this could be a factor playing a role in increasing suicide risk among those individuals that were abused. So how do we study this? We worked in close collaboration with the coroner's office in Quebec, whereby we have access to brain tissue. We run a brain bank that after we collect the brains, we engage in detailed interviews with families of individuals who died by suicide, as well as individuals who died by other causes. And these interviews provide us a lot of information about these individuals. One of the things that we collect systematically is information on their early development. And we collect information on histories of abuse through an instrument known as the SICA that has been adapted to the context of this sort of work. And in addition, we get access to medical files, we get access to files from social services. So in Quebec, individuals that were exposed to adversity and were signaled to social services, they are placed in foster homes, and they keep files, so we have access to those files. So for the study that I'm going to be discussing with you, basically we focused on individuals that had a history of extreme adversity early on in life. They were placed in foster home, they were abused physically and sexually, and the scores were the maximum in the instrument looking at history of adversity. So the glucocorticoid receptor, as I told you, the gene that codes for the glucocorticoid receptor is a complex gene that has 11 untranslated exon 1 variants, and one of these variants, the 1F in humans, it's the homologous to the 1,7. It's a variant that plays an important role in the expression of this gene in the hippocampus. So so we looked at the expression patterns of this variant as well as the full gene, and this is an area where this transcription factor that I mentioned to you in rats binds, both in humans and in rats, and activates this gene. Okay, so when we look at the hippocampus from individuals who died by suicide and had a history of adversity, what we saw was that they had lower levels of expression of the 1F variant, as well as lower levels of expression of the full-length glucocorticoid receptor in the hippocampus. And this is the dentate gyrus of the hippocampus. Oh, this is giving me a hard time here. Right? Okay, so when we look at levels of methylation, what we saw was that individuals that were abused and died by suicide had much higher levels of methylation throughout the 1F promoter, but in particular in one area of the promoter that was precisely the area where this transcription factor binds. And I told you this transcription factor drives the activity of this gene. So we looked at in cellular assays to see if indeed this could explain a alteration of the function of this gene, and we saw that yes, when you methylate that particular area artificially in cell lines, you lead to a decrease in the activity of the gene, and as well this is explained precisely by a prevention of the binding of the NGF1A. So these basically show that that indeed, a history of adversity is associated with increased levels of methylation in this gene, and that this drives or impacts the activity of this gene. So since we've done this study, we look at other variants as well that are important in the expression of this gene in the brain, and we saw that not only the 1F, but as well 1B and 1C and 1H are differentially expressed and methylated according to early life adversity in the hippocampus, but not in other brain areas. Yes, so not in the anterior cingulate, for instance. That's an important area in the regulation of mood. So since we've done these studies, a number of other groups have looked at similar effects in the 1F, and there's very promising data, although still early. We published that initial study in 2009, and there's data coming from a number of other groups looking at 1F in relationship to early-life adversity that is consistent. For instance, here there's a study in a cohort study in the US looking at 1F in peripheral tissue and in relationship as well with the expression of GR that see the same results. There's data looking at even prenatal stress, looking at similar results and so on and so forth. So we are very encouraged by these other results that seem consistent with our initial findings. That said, we need to be cautious because it's still early to say about anything about that. So, oh, come on. All right, so since we've done that, so that was a hypothesis-driven study, so we asked the question of whether or not maybe other genes may also be differentially methylated according to a life experience. So we've done a study at the genome-wide level using an array-based technique where all the promoters are represented, and use an MDEEP strategy, which is an antibody that immunoprecipitates, so basically an antibody that goes and looks at areas of the genome that are methylated and identifies those areas, immunoprecipitate them, and hybridize an array. After that, so when we done this study, what we saw was that 355 different genes were differentially methylated according to a life adversity. Here again, looking at the hippocampus, the distribution of differentially methylated sequences is not specific in the genome. So here you have the different chromosomes, and they are equally distributed in the genome. So which is reassuring, and and it's different from what often we see in cancer, that it's also a phenomenon heavily investigated according to methylation, and where we tend to see areas of the genome that are differentially methylated. Okay, so we had in the same individuals data from expression, so data on function of genes in the hippocampus produced with expression arrays, and then so we contrasted the data from the methylation with the expression, and here what you see in red is data from methylation, in green data from expression. So as expected, the more methylation you have, the less expression you have of that particular sequence or gene. So, and this was across the genome. As you see here, I don't think I've ever had a result that was as significant as this one, when 10 to minus 247. So that's really, really significant, yes, showing that indeed, methylation has a very important effect on inverse correlation with expression. All right, so what did we see on this genome-wide study was that there were a number of different genes, and we won't have time to talk about them. What was also very reassuring is that when we used that genome-wide strategy, one of our top genes that was differentially methylated was also the glucocorticoid receptor. So, so here I'm showing you that either looking at this as through a candidate gene approach or through a non-hypothesis-driven genome-wide study, we end up with this gene coming up. And when we look at all these different genes grouped according to function, what we see was also very interesting. Is that when we classify the differentially methylated genes according to function, we have one function that is, genes involved in neuronal plasticity. that are, among the 5 groups that were the most commonly represented in the genes that were differentially methylated. So basically, I'm telling you here, we're looking at differential methylated associated with early life adversity, and so it makes sense that among the function of the genes that are differentially methylated has to do with neuronal and cell plasticity. Now, we talked about this yesterday, that these epigenetic processes are supposed to be tissue and cell specific. Yes, the study that we did looking at the hippocampus was with homogenates of brain tissue. So we didn't know whether the signal that we were getting was coming from neurons or was coming from other cellular fractions. So what we did was use a technique known as FACS, or a fluorescence-assisted cell sorting, that allow us to separate the DNA coming from neurons from the DNA coming from other cell fractions. So here you have, this is from the hippocampus, this is the neuronal fraction, and this is a non-neuronal fraction. And when we do that and look at the results that we had in the array, basically on the genes that are involved in neuronal plasticity, we see that most, and what, won't have time to go over this. Sorry, I'm having, it's playing tricks on me here. Okay, so that most of the results are coming from really the neuronal fraction. Now, we didn't look at everything, but what we looked at of the genes that were the most, No, the problem is to advance the slides, yeah. Okay. So the of the genes that we looked at, all of the signals that we got were coming from the neuronal fraction. So that was very interesting as a result. Okay, so how can we put all these together from the molecular part? Okay, so we have to acho que a pilha deve estar fraca. Okay, so what basically I told you is that early life adversities associate with methylation in the glucocorticoid receptor. So what it does, the increase, higher methylation in the glucocorticoid receptor, lower levels of expression of that gene. So here you would have a situation with exposure to early life adversity, and here just normal development. So more methylation, less GR in the hippocampus, and in normal situations, you would have more GR in the hippocampus. So, normally the HP axis reacts to stress by releasing cortisol. The release in cortisol gives a negative feedback to the hippocampus, and because you have less availability of GR receptors, you're less capable of having a negative feedback on the HP axis. By doing so, basically what we believe is going on is that this leads to an overactive HP axis because you are less able to negatively regulate the activity of the HP axis because you have less availability of receptors in the hippocampus. So that's the working hypothesis that we have as to how is that methylation early on or adversity early on in life correlates with increased methylation and regulation of genes that play a critical role in the plasticity of systems that are involved in response to stress. Now, we think that these are processes that are evolutionary conserved. We've done studies looking at comparing humans with rats, and this is it's a busy slide, but basically you have all the sequences around the GR gene. You have in the upper panel you have humans, in the lower panel you have the rats, and here you have both. And basically we have a lot of conservation in patterns of methylation across the GR between the species as a result to early life experience. So basically what we think is that the way this works is that your brain gets a signal. I mean, you are, early on in life, you are adapting the tone of systems that respond to stressful events or help you adapt to the environment. If you are in an environment that is very hostile, yes, you need to adapt your stress response systems to react quickly, yes, to be always alert, yes, because you don't know what's going to happen. So, and this is what we think is that these are mechanisms that help the organism better adapt to the environment in environments that are hostile. The problem is when this individual goes from a hostile environment to a non-hostile environment, those adaptations that may be helpful at the hostile environment become maladaptive in other environments. So if we think that this is the case, then being alert, yes, from the clinical point of view, correlates with anxiety traits. Yes, so an anxiety situation, what it is, or being anxious, basically what it is being overly alert. Yes, and being alert. So as I told you, it's a way to defend yourself. Now, to test if what I'm telling you is correct, so we've studied a cohort as representative of the general population from the province of Quebec. So these are kids that have been recruited at the age of 6 and have been followed every year from each until now. Well, every year from age 6 to 12, and then every 3 or 5 years until now they are at the age of 30. They were representative of all the children that attend French kindergartens in Quebec. And from the age of 6 to 12, as I told you, they were assessed on a yearly basis through interviews with teachers and the mother. And they were rated by a number of different behaviors related to development, related to anxiety traits, as well as adversity and traits related to aggression and hostility. So when we look in this cohort, basically children that were abused during childhood, or individuals that were abused during childhood, are more at risk of manifesting suicidal behavior at the age of 21 years. So this looking at 2 points. What we see, and the beauty of these cohort studies that allow you to obtain longitudinal data, yes, and by obtaining longitudinal data over every year, you can draw trajectories, yes. So, with looking at trajectories of development in these kids of anxiety traits, what we see is that these kids basically are divided in 4 different groups. You have the kids that were very anxious at the age of 6 and remain very anxious at the age of 12, and then you have those that were not at all and decreasing and increasing trajectories. Now, membership, yes, in these high anxiety trajectories, once you account for all other factors, leads to an increase in risk of suicidal behavior at the age of 21 of about 60%. Yeah, now, I don't have the data here, but if I did, I would be able to show you that these kids also are likely to remain in high anxiety trajectories until the age of 20. Now, another piece of information that it's important so that you understand the next slide is that one of the other behavior risk factors that play an important risk in increasing suicidal behavior are high levels of impulsivity, and aggression. So this is a study looking at depressed suicides compared with depressed controls at levels of, this is aggression and this is impulsivity, but there are lots and lots of data that suggest that having high levels of these traits increases risk of suicidal behavior. So when we put all this together in this cohort, what we see is that children that were exposed to adversity early on in life and then manifest suicidal behavior at the age of 21. So this relationship between adversity and suicidal behavior is fully explained by development of high anxiety trajectories in kids that also have the presence of impulsive aggressive traits. So those kids with externalizing disorders, yes, this relationship is fully explained. Whether in kids that don't have externalizing disorders, the relationship between having been abused and manifesting suicidal behavior, it's significantly explained by the development of high anxiety trajectories, but not fully explained. Okay, so how can we speculate that this increased in anxiety trajectories as a result of early life adversity and what I told you about regulation of stress response might help us understand risk of suicidal behavior? So, and this is really a speculation, but it's based on data from the literature and data that we have produced in other studies that may help us understand how is that these factors play a role in increasing suicidal behavior. So one of the things that we think is that this HP axis hyperactivity would change, act in outer modulation of value attribution of emotions. So that these outer regulation of how we interpret and put value to emotions basically would lead to impaired decision making in certain situations, and that may be at the core of how people act on these situations and manifest suicidal behavior. So there's data from the literature that clearly suggests that individuals who manifest suicidal behavior had impaired decision making. This data from Fabrice Jolin. And what we've done to look at this relationship is use a paradigm that is known as the tier stress test, looking at relatives of individuals who died by suicide and themselves had been exposed to adversity early on in life. And what we saw is we used two neuropsych tests that look at executive function. And what is interesting here is not actually how people perform in this test, but rather how is that the stress, and maybe I should explain to you in case you're not familiar with the Trier stress test is a test where individuals in the lab are exposed to a psychosocial stress that activates the activity of the HP axis. So basically we invite individuals to, we tell them that we're going to do a test and that they have to tell us about their lives, and then when they're doing so, they tell them, well, hold on, now you have to do a math test, and the math test, it's a very difficult one that nobody can actually do it correctly, and they start making mistakes, and that is very stressful. Yes, so after the test, we debrief them and explain that it was all part of the test. But it is, it's a stressful situation that activates stress response systems like the HP axis. What is interesting is that relatives of individuals that died by suicide and themselves have been exposed to adversity react to the stress very differently than controls, and basically have a hard time adapting and learning from the situation as the controls do. So when you put all these together, yes, we basically think that adversity may be working through plastic regulation of systems that are important of how we react to stressful situations. So these, we think, are mechanisms that help the organism better adapt to the environment, to environments that are hostile. But then when these individuals are moved from a hostile environment to a less hostile environment, these mechanisms that are there to adapt them to better cope with the stress at that situation become maladaptive. And then in the context of all the problems that I didn't have time to discuss with you, may lead to an increase in risk of suicidal behavior. Particularly, one of the factors that I didn't have time to discuss with you that obviously plays an important role is the presence of psychopathology. So, not only, the traits are important in the modulation of how we respond to stress, psychopathology acts as precipitant, yes? So oh, come on. So, we talk about, yeah, I'm almost done. We talk about, early life adversity and how early life adversity through epigenetic factors may regulate the, how behavior develops, yes? So I told you, I gave you the example of development of anxiety traits. Now, then, in the context of other proximal factors like psychopathology, these may put individuals more at risk of dying by suicide. Yes? So I know this is all right. So that's what I wanted to discuss with you. It's a little bit of how we can try to understand the environment playing a role in our biology, and then and how these may play a role increasing predisposition to psychopathology, and particularly here to suicidal behavior. So the work that I discussed with you, it's work primarily, this be from my lab, that was primarily done by Benoît Labonté, who did the work on the arrays and the that I presented on the second part. The work done on the GR was done by Patrick McGowan, who is not in the picture here. And I want to acknowledge as well the work of Naguib Meshawar, with whom I share all the responsibilities on the brain bank. And Yelena Brezo, who did all the work on the cohorts. Yelena is neither in this picture, she had already gone to the lab. And finally, besides people from my lab as well, the work is in collaboration with Richard Tremblay at the University of Montreal, Frank Vitaro and Brigitte Vanner, with whom we've done the work in the cohorts, as well as Moshe Schiff, Michael Meany, Michael Hallett, and Matt Sutherman, who've done, Matt in particular, who did all the bioinformatic works on the arrays. So I guess that's it. Thank you very much.
So thank you, Dr. Gustavo. Since we are quite late, just one short question. Is there any question?
Thank you for the excellent talk. I was just curious about if you have looked at gender differences in the impact of adversity and methylation patterns, because
yeah.
that'd be very.
So that's a great point. We haven't, yes, and the reason for that is primarily a logistic issue. So suicide occurs 4 times more frequently in men than women, yes. So we have an excess of representation of male brains in the bank than female brains. So, these post-mortem studies are difficult because you have to control for so many tissue variables, in terms of the quality of the tissue, that at the end, once you control for post-mortem interval, you control for pH and you name it, yes, and then age of the individuals. Age is a very important factor looking at methylation, and then psychopathology and substance. So when you go from 3,000 brains, you end up with 15. Yeah, once you get everything under control. So we try to minimize variability, and therefore, we decided only to look at male brains. So there you go.
Okay, thank you, Dr. Gustavo. If you want to continue discussion during the coffee break, we are having. Really? Okay, nice one. So sorry. Okay.
Thanks a lot.
Nice question.
Congratulations, Gustavo. Very proud of being on your work. I've been following a little bit because so many things to read. I cannot read everything. So we are proud to have you there and doing such a good work. Trying to learn with you, and I wonder if you could say a few words about the role, how would you, in this puzzle, trying to understand the relationship between child abuse and behaviors such as anxiety traits and things that we are studying, how you put together the immune system. Now we know more and more the bidirectional relationship with the immune system and the central nervous systems. We have now this idea of glial cells, astrocytes, tripartite synapses, and the role of immune cells playing a role in development and brain plasticity. We know this relationship between the immune systems and the HPA axis, inflammation as part of our the understanding of the several diseases that we are studying. We're talking about high levels of corticosteroids. What is the role of the immune system in this model that you are presenting here?
Yeah. So, well, obviously the immune system plays a very important role or more and more we're learning about, its important role in terms of regulation of mood, yes? So yesterday I presented some work that we've done precisely looking at this, yes? So we came across data suggesting that in a subgroup of individuals who die by suicide, we have a dramatic downregulation of astrocytic genes. Yes, and this is very, very consistent. And we've been working on this for about 7 years or 8 years. In the beginning, I thought I had to argue with the trainee that was working on this project in my lab for months, because I always thought that we were looking at byproducts of the collection of brains. Yes, so that I thought that we were looking at factors that were associated with method of death rather than with psychopathology. But he managed to really convince me that we were not, and in fact, because we saw a very dramatic downregulation of genes that account for communication between astrocytes, yes, and then eventually we ended up getting access to a lot of data that suggests that there is indeed a shutdown of a astrocytic network, yes, in the brain of a subgroup of individuals who die by suicide. It has nothing to do with any other factor. So we've been studying this, and we've done, we just completed a study looking at the role of methylation factors, methylation in these results, and we see that it may be an important factor explaining what we don't know very well, and I'm sorry, I'm maybe not very clear. I just don't want to get into a lot of details, but what we don't know very well is to what extent this acts as increasing predisposition, or to what extent this is more proximal, yes? So this is something that we have to figure out, but clearly, if I can try to be clear to, answering your question, is that these factors do play a role also in helping us understand how the immune system, how the glial cells, and in particular astrocytes, may act in regulation of mood or other psychopathological factors. So is that what you asked? Yes.